These are my disclosures for this morning. I first wanted to set the scene by reminding us of the rationale for the recombinant clotting factor platform. These are actual ideas that you can find in the literature if we go back to the late 80s and the early 90s with regards to the development of recombinant technology and how they would be applied to hemophilia. There's ideas that clotting factor replacement products would allow for safety beyond their plasma-derived counterparts, that the consistent manufacturing and processing would be liberated from the uncertainties of securing source plasma. There was potentially unlimited supply that could drive down costs of replacement therapy. This could help facilitate an increase in the utilization of prophylaxis, and that there would be a wider availability of replacement products for patients in developing countries. Now, fast forward 25, 30 years, we could have some discussion and maybe even some argument about how well some of these aspirations have been achieved. In certain parts of the world, they have been realized. In others, maybe not quite as well. But for the most part, many of these are actually the reality of the recombinant era. But maybe what we're realizing now is the ultimate leveraging of the recombinant platform through bioengineering to advance novel therapies. Now, we've been in the recombinant era now for more than 25 years, and it hasn't been without its challenges. If we just look at the biochemical characterization of some of the recombinant molecules we've had challenges with altered post-translational modifications, differences in glycosylation, phosphorylation, sulfation, evidence of dysfunctional proteins within the recombinants with reduced or absent activity, reduced or absent binding to their protein partners. We've been dealing with assay discrepancies, whether you compare against plasma-derived or whether you're using one-stage or two-stage chromogenic assays, discrepancies between the vial content and the labeled potency. And then when we shift to the clinic, we've had examples of altered pharmacokinetic parameters with reduced recoveries, shorter half-life, changes in the volume of distribution. These aren't characteristic of every recombinant, but we've had examples where each of these have been true at different points of development. Reports of reduced efficacy from patients, evidence of increased inhibitor risk through a multiplicity of different studies, and even reduced efficacy in immune tolerance induction. And then if we look at the economic impact, costs have been increased. That's certainly true within the US, but maybe not in other parts of the world, actually. Through tender processes, the costs for clotting factors have been driven down considerably from where they were even a few years ago. Yet we still have restricted access problems in a number of different markets. So despite all of these challenges in the recombinant era, I would tell you that every one of my patients are treated with a recombinant product in my clinic. Recombinants have been advocated for by almost all of the bodies that provide advice for therapies. So the reality is we seem to be able to overcome these challenges, and I think that will also be true when we're talking about bioengineered molecules. Almost certainly when we change the physiology of these molecules, things are not going to be the same, and we will have to adapt and accommodate to those changes. So what is the promise of bioengineering that could eliminate some of the remaining gaps for our patients? We still want to reduce and eliminate the barriers to effective prophylaxis, the treatment burden, the IV access challenges, the costs. And now as we move firmly into the gene therapy area, the issue of expression has become more and more important. The higher levels that we can achieve, the better outcome for patients, and also the less aggressive we have to be with the vector transfer to get those levels of expression. We would love to be able to eliminate inhibitor risks definitively, and it would also be nice to reduce some of the interpatient variability in pharmacokinetics through bioengineering strategies as well. Now, the process of bioengineering recombinant clotting factors on this platform, we've learned much from basic research insights, studying the structure and function of factor VIII and factor IX across their life cycles, biosynthesis, macromolecular interactions, activation, inactivation, clearance. All of these have given us insight of where the targets are for modifying these molecules. It's interesting that the study of hemophilia causing mutations, obviously Padua is not an example of this, but both studying hemophilia mutations and just patient mutations in general have given us a lot of insight of how to modify molecules and produce improvements. Site-directed mutagenesis is where we either do random mutagenesis of the molecule or rationally make changes informed by some of the studies I've talked about. We've had detailed structural models that have given us insights on the folding and the 3D structure of the molecule, but also its intermolecular interactions, and that's given ideas for new targets. And then we've had a wide repertoire of animal models to test these in long before they make it to the clinic. There's a number of bioengineering strategies that have been used to enhance biologics in general, but many have already been applied to factor VIII and factor IX. We won't go through all of these, but there will be a few selected ones that we'll talk about. But just in general, in many cases, we are modifying the cDNA. We're doing things like B-domain deletion or truncation, amino acid substitutions. We're changing interactive sites. We're introducing novel conjugation sites so that additional conjugates can be placed onto there. We're targeting immunogenic epitopes. We're playing with single-chain molecules to see if that changes the properties. We can alter the post-translational modifications. Sometimes is through introducing novel glycosylations, but sometimes just simple choice of cell line for expression can have differences in the product. Adjusting the production efficiency and consistency to reduce aggregates, optimizing the post-translational modifications. The polymer conjugations have been well vetted in this community with polyethylene glycol conjugates. We heard at this meeting about X10 as a novel conjugate as well. And then the fusion proteins, the FC fragment and albumin, are now well established within the clinics. If we look over the current and future approaches of care, within the replacement therapy eras where recombinants first had their opportunity for testing in the clinic, and then they've been further modified to advance in the prophylactic era, moving from the standard half-life to the extended half-life. The non-replacement therapies are not going to be the focus of this morning's talk, but these have also been well vetted even at this meeting, and also are evidence of application of bioengineering. But really it's related to prophylactic therapy and within gene therapy that we're really seeing leveraging of bioengineering resulting in significant improvements for our abilities to advance these therapies. So I'm going to use this, this graphic as sort of a layout to describe a few of the examples of bioengineering and how they impact over the life cycle of factor VIII. So basically we're moving, imagine this is either a cell for cell line production or this is a target cell for gene therapy. Ultimately that DNA sequence has to make it into the nucleus. A message is created from the cDNA. That message has to be delivered to the rough endoplasmic reticulum where those ribosomes translate that into protein. Within the endoplasmic reticulum, those proteins fold. They acquire many of their post-translational modifications, and then they move from the ER into the Golgi. There's additional processing that occurs in the Golgi, and then ultimately they're secreted, and in some cases, as you'll see with factor VIII, have to be stabilized through other macromolecular interactions to ensure its proper function. So Thierry introduced yesterday this idea that factor VIII production particularly has been a bottleneck for production. And this has been a source of study and an interest of mine and many others for many years, is why is factor VIII expression so inefficient? And it does start right at the messenger RNA level. Factor VIII has a challenge in producing messenger RNA. There's sequences within the coding region of the full length that have a deleterious effect on messenger RNA accumulation. There's sequences that have been identified that can lead to transcriptional silencing. And then even if that message gets delivered to the ribosome, you have to be able to translate that protein efficiently. And we'll just mention a couple here that I'll go into, and it's the B domain deletion and codon optimization that have had the biggest impact in this part of the biosynthetic cycle. So we've been talking a lot about B domain deleted factor VIII at this meeting, actually. So what is it that we've actually done to the molecule? This is the predicted primary structure of factor VIII, these triplicated A domains, these duplicated C domains, and then this large central B domain, as well as these acidic regions that are indicated in white. But that's not the whole story of factor VIII that's derived from the cDNA. There's a number of post-translational processing of factor VIII that decorate the molecule in different ways. Many of these are derived in the ER with signal peptide cleavage, these asparagine-linked glycosylations that are decorated across factor VIII, disulfide bond formation, copper ion binding. And then as it moves to the Golgi, there's a number of other changes, complex changes to the sugars, tyrosine sulfation, and then ultimately heavy and light chain cleavage. But very early on, after the gene for factor VIII was first cloned, it was determined that this large B domain could be deleted. Why did they even choose to do that? Well, when they started looking at other animals, other animal species of factor VIII, they noted that there was significant divergence in the sequence within the B domain, whereas there remained high homology in these A domains and C domains. And when they removed that B domain, they were able to show that it really was dispensable for procoagulant activity. This was the very first bioengineered factor VIII, and it goes all the way back to 1986. And 30 plus years later, we're now using this regularly in the clinics, and it's helping advance gene therapy for factor VIII. What about codon optimization? So what is the principle of codon optimization? Maybe this hasn't been a focus of this meeting in particular, but this is the genetic code. These triplet codes that are derived from the cDNA and incorporate into the message are what help guide the tRNAs to bring the appropriate amino acids when the protein's assembling. And the genetic code is degenerate. So there are multiple options, different, if you like, DNA sequences that can code for the same amino acid. Except for methionine and tryptophan, all of these have multiple triplets to code for. So the principle with codon optimization is that some codons are rare and they differ between species. And if you replace rare codons with ones that are more frequently used, you can actually increase protein expression. So the idea is that these rare tRNAs limit the efficiency of the protein expression. Now, codon optimization has actually been used for one commercial extended half-life factor IX for regular infusion, but it's the mainstay of factor VIII and factor IX transgenes for gene therapy. And by adding codon optimization on top of B domain deletion, this really substantially increases the protein expression. And it's allowed for us to reduce vector dosages and achieve higher plasma levels. But what does it mean to do codon optimization? There is a wealth of software programs that help design and do this codon optimization in silico, but almost certainly they don't use the same algorithms, and it would be very unlikely that any two codon optimized molecules would be exactly the same. Now, I showed you the degenerate code, and you could say, well, what is the big deal? If it ends up with the same amino acid sequence, this really shouldn't have any implications. But turns out there are unanticipated effects of codon optimization. There can be protein effects, altered protein conformation and stability, altered post-translational modifications, even altered protein functions on the activity, interaction with drugs and inhibitors, phosphorylation profiles, sensitivity of the proteins to proteolysis, or even the propensity for aggregation. Some of the proposed mechanisms include that codon usage actually determines the translation rhythm. So this causes the ribosomes to slow down or pause at certain sites because of these rare triplets. And that these pauses actually modulate the sequential folding events that occur co-translationally. So this really acts as a secondary code that's guiding in vivo protein folding. So what's the evidence that this really is an issue? Well, again, we learn from our patients. So this is a mild hemophilia B mutation. It's a single base change. And you can see that it's what we would call a synonymous mutation. The amino acid sequence for this patient is identical from their peers. The valine at position 107 remains valine, but there is this base change. And this patient has a factor IX activity of 15 to 20%. So with the native codon, that gets translated ultimately into a protein, and we would end up with the normally correctly folded factor IX. However, with that single base change, this mutant codon, first of all, it can alter the mRNA structure. So we don't often think about the structure related to messenger RNAs, but they're not just long strings. They actually have a secondary structure. And the idea is that there may be some altered kinetics of translation related by altering the rhythm of translation that occurs at the ribosome. This can alter the protein conformation and processing. And with most things in an assembly line, if things start to get backed up, you have to get things off the assembly line. And so what this happens in the cell, you start to accumulate proteins intracellularly, you move them to the garbage cans, and so you can get enhanced protein degradation. And so ultimately, with this single base change but a synonymous mutation, we end up with reduced protein expression and secretion. So I guess the point of this is that related to codon optimization, this is maybe yet an unexplored evaluation of what we're doing as we head into the gene therapy era. So what about codon optimization specifically of factor VIII? Well, some preclinical studies have shown us that codon-optimized B-domain deleted factor VIII exhibits a 7-fold higher expression from within CHO cells, but there were some observed differences in post-translational modifications, both N and O-link glycosylations, the degree of tyrosine sulfation. And interestingly, in this one example, the specific activities was 1.5-fold higher by a one-stage clotting assay compared to chromogenic. Now, that should strike you as odd because what we've always seen with B-domain deleted factor VIII is that the one-stage activity has been up to 50% lower than the chromogenic. And this might not have had the same significance until we saw data from a recent clinical gene therapy with B-domain deleted factor VIII, in which the same phenomenon was seen, where the one stage was about 1.6 that of the as measured by the chromogenic. Again, not predicted by what we've known about B-domain deleted factor VIII to this point. We don't have any insights of whether this is actually the mechanism that's involved in both of these instances, but it does sort of highlight that when we do codon optimization, this is a software mechanism, but it isn't necessarily informed everything about the biology of this so-called secondary code. So now we need to move that protein through the ER, and one of the challenges initially, particularly for factor VIII, is the efficiency of protein folding. Factor VIII has a number of challenges. It tends to accumulate within the ER. Because of that, it interacts with these ER stress protein chaperones. Because of all those decorated glycosylations, it interacts with a number of other chaperones that are recognized unfolded proteins that have those N-link structures. And it's also in an oxidating environment. Disulfide bond formation contributes to oxidative stress inside the ER. And there have been some innovations that have helped overcome some of these protein folding challenges. I won't talk about these in any detail, but sometimes single mutations can alter the interactions with things like this BIP protein. And at least in one example, in a cell culture expression for a commercial factor VIII, co-expressing with a heat shock protein was used to enhance the production efficiency of the cell line. Then we have to move the protein from the ER to the Golgi. And we really didn't have any insights on this for a long time until we started studying patients. And patients with the rare bleeding disorder of combined factor V and factor VIII deficiency were an enigma for a number of years until it was recognized that all of them had mutations in this complex, this LMAN1 CFD2 complex. This is a transmembrane protein, and it includes a mannose-specific lectin, and it forms a calcium-dependent heterodimer. But what it does is it seems to recognize heavily glycosylated proteins and really acts as a shuttle mechanism using a facilitated transport mechanism to move them from the ER to the Golgi, and has evidence that it interacts specifically with factor V and factor VIII. So why would this be the case? Well, we go back to our structures. This is to remind you that factor V is significantly homologous to factor VIII, both in its assembly within the enzymatic reactions, but also in its overall structure. And then you see both of these B domains have all of these clusters of N-link glycosylations across the B domain. So we had studied this a number of years ago. What happens when you delete the B domain? You lose not just the B domain sequence, but you also lose all those N-link glycosylations. And we had observed that when you delete the B domain, you get that boost from the message, but you don't necessarily get that much more protein out of the cell. And so the hypothesis was, did we once we understood this facilitated transport mechanism, were we losing something by not having at least some B domain in there to help take advantage of this facilitated transport? So we looked at a number of constructs adding sequentially N-link glycosylations and native sequence. And we actually found that with about 226 of amino acids that include 6 N-link sugars, that we could substantially boost the expression of factor VIII. And this showed increased messenger RNA, improved ER Golgi transport, and actually showed evidence of reduced ER stress in cell lines in animals. And this has been now looked at in a number of preclinical in vitro and in vivo applications, and it has replicated what was seen in the cell cultures, about a 10-fold boost in expression. Now, the challenge with this construct is now that we're on the AAV platform for gene therapy, this is just too big. Factor VIII is already at the cusp, barely, to get it packaged within the AAV capsids. And so the group from this publication took the insights on the N6 molecule, and they tried to keep the size of the B domain deleted construct the same, but introduce through a couple of different variants N-link sugars to take advantage of this facilitated transport. And they had 2 variants. We'll focus on the V3 variant, because when this was moved forward in preclinical studies in the murine models, it actually substantially improved the expression of the B domain deleted factor VIII just by introducing these few engineered N-link sugars. And that has now moved forward. The GO8 study you may be familiar with is a phase 1 hemophilia A gene therapy, and it's using this variant 3 factor VIII, which has these 6 N-link sugars to help with the overall expression. Now, we can also do bioengineering through comparative biology. And this is of some interest. So I mentioned comparing against other animals previously. So if you look to porcine factor VIII, we actually now have recombinant B-domain deleted porcine factor VIII. This is expressed about 10 to 100-fold higher than human factor VIII. And it shows decreased interaction with ER chaperones. It shows that it has an attenuated unfolded protein response. And it seems to be related to sequence within the A2 domain and the activation peptide prior to the A3 domain. And so a porcine human hybrid that was termed ET3 basically exchanged about 9% of the sequence of the human with porcine in these areas. And what they achieved were an 8-fold reduction in the AAV vector dose that was needed to achieve therapeutic levels in mice. So again, a nice partnering between bioengineering to help improve the efficacy and perhaps safety of AAV applications. but we also learned from canine factor VIII. So human B-domain deleted factor VIII is expressed as a heterodimer, but the canine is primarily a single chain. It has a higher specific activity, has higher stability, and also it is believed to have reduced cleavage by this processing enzyme called furin, which processes the protein in the Golgi. And so the group that was studying this engineered B-domain deleted factor VIII with modifications to the cleavage site, modifying it to that of the canine sequence. And then what they were able to observe from the Penn group's work is that they had a twofold increase in expression in AAV gene therapy in dogs. Now you can extend this further with what would be called ancestral comparative biology. And this is work from Phil Zakas, who's currently at Emory. And what he's done is sort of reconstructed the ancestral differences between factor VIII across a wide range of animal species. And then you can start to look at, when you express those variants in cell lines, you can look at what are some of the differences in expression that are observed. And you can see there's wide ranges of efficiency across these different species. And by going back and looking at where the amino acids begin to change and diverge across this comparative biology, you can do targeted mutations in the human and try to translate those improved expressions into the human construct. So finally, we end up at the far side of the biosynthetic pathway. And we're still challenged with factor VIII by its binding to von Willebrand, need to bind to von Willebrand factor. So we know that it stabilizes factor VIII on secretion. It prevents factor VIII chain dissociation. It displaces factor VIII from the phospholipid surface, reduces its degradation. And so this is an important part of the biosynthetic cycle. Now, we had studied in partnership with Dave Ginsberg that the von Willebrand factor fragment, the D' D3 domains of VDEBF, were sufficient to stabilize coagulation factor VIII in mice. So when we took a D' D3 domain of VDEBF and fused it to the FC fragment, we were able to show that it prolongs the survival in vivo and elevates factor VIII levels in a VDEBF deficient mouse. But when we took that same construct into the Hem A mice, we couldn't see any enhancement of factor VIII. And it was believed that the D' D3, this fusion fragment, was really ineffectual in competing with endogenous VDEBF. There's an equilibrium, there's an on-off for factor VIII, and so gradually it just migrates to the native VDEBF in the hemophilia A context, and you lose the advantage of that D' D3. So Dr. Frubus introduced us again at this meeting to this innovation that builds on this principle. And so in order to lock the factor VIII onto the D' D3, they used FC fusion of both the factor VIII and the D' D3. So that covalent linkage then eliminates that on-off equilibrium. And they also enhanced the reduction in clearance by introducing these X10 insertions. They reminded us at this meeting that in the preclinical models, this was able to break the ceiling, if you like, that had been challenging the extended half-life factor VIII molecules, increasing the factor VIII to about 32 hours. And then really the wonderful announcement at this meeting was from the BIV001 phase 1/2 program that this did translate into the human experience as well. So 37-hour half-life in subjects with severe Hem A, and at 7 days retaining a factor VIII activity of 5.6%. So it looks like we have been able to break through this ceiling by understanding this biology deeper. Now, we shouldn't forget factor IX. Factor IX Padua has had lots of coverage at this meeting. This was originally described in this family pedigree, who actually presented with a thrombophilic phenotype. And this is a single nucleotide change that results in a substitution of arginine at position 338 to leucine. And this fit with some older preclinical data of targeted mutagenesis in this area of the molecule, that this would have enhancement enhancement of the activity of factor IX. And of course, we've seen how this has helped advance gene therapy for factor IX in one program that's been published, and now really has become now the mainstay platform for factor IX gene therapy going forward. But we also saw at this meeting evidence of targeted mutagenesis of factor IX to change its properties. This molecule has had a targeted mutation here to produce resistance to antithrombin, and then a couple of mutations that alter the affinity for 8A and would increase the procoagulant activity. And this molecule has shown a 22-fold potency advantage over the native factor IX, and when administered subcutaneously, you can see that plasma levels with repeat dosing in this clinical trial are achieving well into the mild range, approaching 12 to sort of 15 or 18%. So again, another example of how insights from the biology has allowed for targeted mutagenesis in order to produce some real novel therapies for the clinic. So in conclusion, rationally designed factor VIII and factor IX variants have been informed strongly from evolving basic science insights on biosynthesis and biochemistry, and from the study of patient mutations. The bioengineered 8 and 9 are the key to unlocking the full potential of gene therapy, with levels now within the therapeutic range and also limiting the vector doses that are needed for efficacy. I think these variants are proving themselves in the clinic. Now, these are all going to introduce new challenges with our traditional bioassays, clinical correlations, unanticipated effects on the cell, biosynthesis, and macromolecular interactions. But I believe that as we've seen already in the recombinant era, we will be able to overcome all of these challenges as well. So thank you very much.